Welcome back.
Welcome.
This is part 2 of the project.
It's Ava and
The
Grace.
progress
Hell's
thing.
Kitchen.
Yeah.
That's our group name.
And we're going to talk about chapter 9, which is physical and chemical control of microbes. So, controlling microorganisms, you see this in everyday life, such as salting foods like salted meats, smoking food like smoked salmon, pickled pickles, and drying food. And like I said, a lot of these examples are in food. Storing water in certain containers, like made out of copper or silver, is another example. So results of microbial control methods. We have sterilization, disinfection, antisepsis, decontamination. So sterilization would be the destruction of all microbial life, including spores. Disinfection destroys most microbial life on inanimate surfaces. Antisepsis would be the same as disinfection, but on skin. It can be used on skin. So, like alcohol and iodine. Decontamination is a more generic term, also called sanitization. So, it's the mechanical removal of most microbes from an animate or inanimate surface. Primary targets of microbial control, just getting rid of as many microorganisms that are capable of causing infection or spoilage out of the environment. So resistance of different microbial types. Endospores and prions are the most resistant. Enveloped viruses are the least resistant, and gram-negative is more resistant than gram-positive. We have that on the flow chart. Oh, CJ is calling me. I cannot take your call right now, love. Already went through that. So, sepsis versus asepsis. Sepsis is the growth of microorganisms in blood and other tissues, and it's usually lethal. So, if you ever heard of somebody's gone septic, asepsis is doing things to prevent the entry of microbes into sterile tissues, and it prevents infection. So, example would be heat sterilizing your tubes, surgeons wearing gloves and masks. Antisepsis is the application of chemical agents to exposed body surfaces to destroy live pathogens and reduce the number of bacteria, but it does not kill spores. So, example, before you get a shot, the nurse rubs alcohol swab on your skin. So, static versus cytal. Static inhibits growth while cytal kills the cell. And we also have bacteriostatic versus microbiostatic. So bacteriostatic is going to inhibit the growth of bacteria. Microbiostatic agents are chemicals used to control microorganisms in the body. So microbes. Yeah. Cytal, germicide, microbiocide kills the microorganisms. It does more than just inhibits the growth. Bactericide destroys bacteria except for endospores. Actual cell death is the microbes die and the cell is destroyed, so you won't be able to see it in the microscope. Mode of action or killing is what targets the major processes of microbes. So common targets are the cell wall, cell membrane, synthesis of DNA or RNA, and the major cellular proteins, so like enzymes, ribosomes, because once the protein shape is denatured, then its function is no longer the same. Yeah. Cell membrane, mode of action of surfactants on the cell membrane. So like classic is sodium lauryl sulfate, which is a component of Dawn soap. It has the highest microbicidal action. It's a surfactant. So the surfactant molecules are the polar molecules with hydrophilic and hydrophobic regions. And so once the surfactant physically binds to the lipid layer and penetrates the hydrophobic regions of the membranes, this opens up holes that allow other chemicals to get into the cell. So this is like soaps, detergents, bile that do this. Went through that. Physical factors. Heat as a killer. Moist versus dry heat. Moist heat would be boiling water, and the best example would be autoclaving. Autoclaving is like probably, you kill, in order to kill spores, you would want to autoclave. So it's 121 degrees Celsius. What?
Used for like all like medical tools
and I'm pretty sure in the lab.
Stuff, right?
Yeah. Mechanism, denaturing of proteins, blows up and kills cells. Pasteurization is also moist heat, but it's at a lower temperature, and probably the biggest example for this is milk. So like pasteurized versus unpasteurized milk. Unpasteurized milk scares me. I don't know why people drink that. And then there's dry heat, which is like really hot, but it'll take longer than moist heat because it's dry, low moisture content. Dehydrates the cell, burning it to ashes. The lack of water increases the stability of proteins. That's why a higher temperature is necessary. So like in the most effective to least effective, it would be autoclaving, then incineration, boiling, and then pasteurization. All of these are bactericidal, kill bacteria. And then on the other hand, freezing, this slows down microbes, so it's a static, not cidal. So we have desiccation, which is the drying, dehydration of vegetative cells. And then we have lipophilization, which is a combination of freezing and drying. So think of freeze-dried ice cream. Let's see. And some pathogens can be killed, but some are dormant. So they're just not like active. But once it warms up to room temperature, like the spores that survive could become active. Radiation is another mechanism of killing. Am I going too fast for you?
No, you're fine.
Okay. Yeah. So irradiation, exposure of microbes to radiation. Shorter the wave, higher the kill. Ionizing radiation would be gamma rays and X-rays, high frequency. So the method is radiation knocks out electrons from atoms, causing ions to form and destroying proteins and other polymers, including DNA. So it'll cause like mutations in the DNA. Non-ionizing would be UV rays, and this leads to thymine dimers in DNA. So this also will mess up the DNA. That's like, yeah. Physical factors, filtration. So you can filter air and liquid. Air filtration would be a porous filter, removes microbes from the air. So like HEPA filters that everybody had after COVID. For liquid, you can have a coarse or ultra fine like filter, depending on what you're trying to filter out. True sterilization would be by removing viruses and even large proteins. So like beer, milk, you can filter. And then osmotic pressure cell shrinks in a hypertonic solution because the water is leaving the cell and it's trying to dilute what's on the outside. So, like when you salt foods, that's what you're doing. You're making